the Academy, the Israel Academy of Sciences, actually it's Academy of Sciences and Humanities. In Hebrew it is called Academia Lemadaim because we have Madai HaTeva and Madai HaRuach, and both sciences and humanities are called sciences. But the topic of the lecture today is one of a few examples in which both humanities and sciences are included in the substance and in the essence of the lectures. And being the president of the Academy, it is really a pleasure to chair a session that brings together the two divisions of the Academy to one scientific occasion that will deal with a very interesting problem. We have a full program, so I will not take any more time, and I will invite Professor Richard Williams, who is the president of the Estonian Academy of Sciences, to say the words of greeting. Please, Dr. Williams.
Good afternoon, everybody. It's a privilege, of course, and pleasure to be here and to say these few greeting words. But what was already mentioned, the science, what would be discussed today, that is truly not just interdisciplinary, but transdisciplinary. What I keep in mind is that my background is medicine. My background is medical biochemistry, molecular biology, switching maybe some 20 years ago more to population genetics, to genomics. But all the ideas in this field come not from that much from genomics as such, or genetics as such, but they come one half because genes are so important for diseases from that side. And the other half, the real questions, what to ask, they actually come not at all from, so to say, American meaning, Anglo-Saxon meaning of sciences. They come from humanities. I mean, so you can do the same amount of work or 10 times more work measured by, say, dollars, or measured by hours, or measured by postdocs, if you want, or PhD students, without much meaning. Just you sequence, pile up genomes, and then comes the question, what is a question actually? Why I really became involved? There's a series of just chances. First meeting was in Mary Gelman place in Santa Fe exactly with the people who deeply studied Semitic languages, the split pattern of the Semitic languages. And they have been among the greatest experts on that. I started to understand that there is something very interesting going on, starting from Akkad language, and then the split of Ugarit and Hebrew and Aramaic and so on, Phoenician. That was one thing. And the second was, of course, the so to say, general knowledge shared by all Western world about the Bible. And the third, of course, was that I met, I think it was Jim Watson's place in Cold Spring Harbor, one of the dedicated meetings on human genetic history, Dororon. And then afterwards, next stage, when Karl, Professor Skoretsky, invited me to Haifa, and we could really, with Bacheva and involved, to discuss what would be the questions to ask. And then it came, and I'm very happy that this is already, say, dozen years roughly going on collaboration. What now told me a very good lesson how I eventually start to learn Estonian genes. I've co-authored probably maybe 5 papers or 6 papers about Jewish genetics. I haven't really managed yet to write any about Estonian genetics, but now I know how to do it. Thank you very much.
Thank you. Thank you very much, Professor Williams, for these interesting words. And I would now like to ask Professor אורי זליגסון.
נטוש ויכוח מדמם, הייתי קורא לו, לגבי המוצא הכוזרי של האשכנזים. וזה מעורר מזה עשרות בשנים ויכוחים אינסופיים של היסטוריונים. החידוש הגדול שקרה וקורה גם בתחומים אחרים, שהגנטיקה נכנסה להיסטוריה. זאת אומרת, אנחנו, למעשה כל אחד מאיתנו שיושב פה, הוא living monument, מפני שהוא נושא באמתחתו, ב-DNA, אינפורמציה של אלפי שנים, ושאפשר להיעזר בה למטרות שונות, למשל כשרוצים לבדוק שאלה כל כך קריטית, האם האשכנזים הם כוזרים כן או לא. ודורון בהר, אני בטוח, יספר לכם הרבה דברים חדשים ומעניינים. אבל עוד קודם לכן, אתם יכולים לשאול את עצמכם מה עומד כאן המטולוג שנושא מחקרו קרישת הדם ומדבר על הדברים מהסוג הזה. ובכן, אני מודה ולא אבוש שמי שגרר אותי לעניין היה מורי ורבי, המנטור שלי, פרופסור חיים שיבה, שהיה לו עניין גדול מאוד בעדות ישראל, והוא וגנטיקאים מפורסמים אחרים כמו אליזבת גולדשמיט, ריצ'רד גודמן זיכרונו לברכה, עסקו במשך עשרות בשנים בגנטיקה של העם היהודי. ואחד הדברים הראשונים ששמו לב אליהם זו העובדה שכשמסתכלים על היהודים האשכנזים והאחרים, הם נראים בכלל מעמים אחרים. תראו את אלה, מבנות ישראל. מה השייכות בכלל לזקן העיראקי, ילד תימני? האם אלה דומים? זה ברור לגמרי שהם קורצו מחומר אחר, אבל אנחנו לא כל כך בטוחים בזה כפי שתשמעו תכף. וכאן גברת מלוב גם נראית אחרת, כמובן התלבושת קצת מסיתה את העניין. וכאן תנחשו מזה, מצד שמאל, זה אני בעוונותיי הרבים, וכאן נמצא הסבא, הילד הזה, שהוא היום סבא, לנכדים האלה, הקטנים ולבת. יש עוד, אבל לא את כולם הרי, כמובן. כך שהמחשבה הראשונה הייתה כשהתכנסו כל עדות ישראל חזרה בארץ ישראל, שפלשו גנים מהסביבה וחדרו לתוכנו. ואין הוכחה מוחלטת שזה כך, אבל נראה עוד כמה עובדות שהן חשובות לזכירה. כאן לפניכם פשוט רשימה של מאמרים מ-1959, שבעצם כשמסתכלים לעומק כל מקרה ומקרה, ולא רק בתווי הפנים החיצוניים, יש דמיון רב מאוד בין העדות ושוני גדול מאוד בין העדות לבין הלא יהודים. כאן לפניכם עבודה של קרלין, של לולק שנברג, זיכרונו לברכה, ליפשיץ, שלקחו פולימורפיזמים, אלה שינויים בגנים, שלא גורמים לאיזושהי פונקציה לא טובה, אלא הם נמצאים שמה, והם מאוד עוזרים לנו בשביל לעשות את האבחונים על סמך הפולימורפיזמים, על סמך השינויים שחלים בפציינטים שיש להם את אחת מהמחלות הנדירות, שאז ב-1959 ו-91 אפשר היה עוד לדבר, או אי אפשר היה לדבר על גנטיקה מולקולרית, מפני שזו עוד לא התפתחה, והסתמכו על פולימורפיזמים על סמך אלקטרופורזות ושיטות אחרות. והדבר השני שראו הגנטיקאים אבותינו, שבין האשכנזים יש מחלות גנטיות בשכיחות גדולה מאוד, וייחסו את זה, מייחסים את זה למספר היחסית קטן של האשכנזים שנותרו בחיים אחרי הדבר השחור, ושלאחר מכן הייתה התפוצצות האוכלוסייה, כפי שדלה פרגולה מספר לנו בהיסטוריה. זה הגורם כנראה לשכיחות של המחלות האלה הגנטיות, ויש היום למעלה מ-40 מחלות אצל האשכנזים שאין בעדות אחרות, וכמובן שזה מכרה זהב לכל החוקרים שרוצים להיכנס לעובי הקורה. אז תראו בין השינויים, וכאן אני חוזר למקצוע האמיתי שלי, בשורה התחתונה, שמחסור בגורם הקרישה 11 שכרוך בנטייה לדמם בעקבות חבלות, שכיח בצורה פנומנלית בין האשכנזים. אחד לכל 11 יהודים אשכנזים הוא נשא הטרוזיגוטי של המחלה. ואחד ל-450 הוא לוקה במחלה עצמה. יש עוד מחלות תורשתיות כאלה, אבל כמובן שזה לא הזמן לפרט אותן. כשהלכו ובדקו מה המוטציות שגורמות לחסר פקטור 11 באשכנזים וגם בלא אשכנזים, נמצא ששתי מוטציות הן דומיננטיות, אלה השתיים בראש הטבלה. יש סוג אחד שהוא טייפ 2, סוג שני שהוא טייפ 3, והשכיחות היא דומה ביניהם, אבל מה שמרתק, ויש עוד כמה קטנות בדרך, אבל מה שמעניין מאוד זאת העובדה שהמוטציות האלה קרו גם בבני עדות אחרות. כפי שאתם רואים כאן, זאת אוכלוסייה נורמלית שבדקנו אותה. הסוג 2 של המחלה קיים, אתם רואים בין ה-531 היו 21, ובין היהודים העיראקים, שכביכול לא צריך היה להיות ביניהם קשר, אתם רואים שהשכיחות לא הייתה מבוטלת, וגם בערבים פלסטינאים, יותר מאשר עדות אחרות כמו צפון אפריקה. מה לגבי המוטציה השנייה? המוטציה השנייה שקראנו לה טייפ 3, אתם רואים שהיא מאותרת רק באשכנזים, ובמאות מקרים שבדקנו, אף אחד מהם לא היה הטרוזיגוט למחלה. כלומר שפה הייתה עוד נקודה שצריך לחשוב עליה כשעושים את המחקר בנושא הזה. ובשיתוף פעולה עם גנטיקאי מפורסם דיוויד גולדסטין, שכיום יושב בדיוק, עשינו תחשיב של גיל המוטציות האלה. ואני לא אכנס לפרטים איך לעשות את זה, זה נעשה על סמך פולימורפיזמים שונים שנמצאים בגנים של החולים האלה, ואנחנו יכולים לסכם היום שהיות וטייפ 2 קיים גם באשכנזים וגם בעיראקים וגם בערבים פלסטינאים, אנחנו מניחים ודייוויד גולדשטיין אישר את זה, שהפאונדר אפקט, המייסד של כל השבט, חי בתקופת גלות בבל. כלומר, לפני 2,500 שנה. ונדד עם הפלסטינאים, העיראקים והאשכנזים. כך שהתמונה היום היא, כפי שאתם רואים בתחתית התמונה. והמוטציה השלישית, יש גם מוטציה טייפ 1, שהיא פחות שכיחה, הרבה פחות שכיחה, היא הטייפ 3 מאותרת ליהודים האשכנזים, כך שהיא קרתה אחרי שבני העדה האשכנזית עזבו את המזרח התיכון וייסדו את הקהילות באירופה, והיא קיימת היום באלה שהצגתי. אז בלי להכביר יותר במילים, המסקנה שאנחנו יכולים להסיק על סמך הנתונים הגנטיים האלה, שהשינויים הגנטיים האלה הם אופייניים לעדות מסוימות, אבל גם בין עדתי, ואנחנו יכולים היום להגיד בביטחון גדול למדי, כפי שאנחנו נשמע בהמשך הערב, שאלה שהם אשכנזים הם באו מהמזרח התיכון, ואין קשר כנראה לכוזרים בכלל. אבל פרטים נוספים תשמעו בשתי ההרצאות האחרות. תודה.
תודה, אורי. ואני עכשיו מזמינה את פרופסור אריאלה אופנהיים מהאוניברסיטה העברית, להגיד גם כן כמה מילות פתיחה.
Well, our interest has in genetics of populations, communities, etc., started when from our interest, really emerged from our interest in ancient DNA. About 15, 20 years ago, we found in old bones that were found in this country, prehistoric bones, about 10,000 years old. We found several types, and this was very, very general, not refined study, but we already found several types of polymorphisms. That told us that also in the prehistoric times, the population here was heterogeneous from the genetic point of view. And in order to understand the evolution of the population in this area, we decided to look at present populations. And our first study was on several Jewish communities and Palestinians from different parts of the country. And to our amazement, we found that they belong to the same genetic pools. We really did not expect it about 15, 16, 17 years ago. Maybe today it sounds very common sense, and we all know it, but believe me, at that time, it was really news. And even in Al-Quds, the newspaper that is published in East Jerusalem, the title was, "We are not cousins, we are brothers." So another amazing thing that came from that old study that was very crude in terms of polymorphisms and in terms of techniques that were available at that time was that there was a genetic continuity, an amazing genetic continuity along 2,000 years that we really did not expect to find. And Sephardic Jews from North Africa and Sephardic Jews from other places like Iraq or Turkey, Greece, were really identical, the same genetic gene pool. And I must just limit it a little bit because we just studied the paternal chromosome, the Y chromosome that is inherited from father to son. And Ashkenazi Jews were slightly different, but altogether Jews and Palestinians shared a very large of their genes or of their Y chromosomes came from the same branch. So this really told us, since we are dealing with males, that there was no really major conversion from other populations into Judaism. Of course, we studied only Jews, so we couldn't follow conversions of Jews to other populations or communities. Then, a few years later, in that particular study, we already detected that Ashkenazim and Sfaradim were a little bit different. And the difference was in a single branch that was not present in Sfaradim, only in Ashkenazim. So later we started investigating this branch and found that it is present today in Eastern Europe, very widely spread there, and also in large amounts. So the question arose, how did it get to the Jewish population? Using all sorts of genetic technologies, we came up with the conclusion that they entered the Jewish population in the first millennium, and since the source was in Eastern Europe somewhere, we weren't quite sure where, we put up the hypothesis that maybe it represents the Kuzarim. Now, here, I don't know about you, but I remember this book that we studied at my time, my old time, we studied in high school. It's an amazing book. It's a philosophical, really a philosophical composition written by Yehudi, and it is based on the story that the Kuzarim were converted to Judaism. I don't have to repeat it. So we put up the hypothesis, and we said, we took several samples of Ashkenazi Jews from different communities in Israel and other countries, and in all the communities, the percentage of this particular chromosome was about 10–12%. So we said it could be the Kuzarim, and if it is, then the Kuzarim contributed to present Ashkenazim not more than 10–12%, and unlike many other historians, they talked about 80–90%. So I'm sure that a few years later, well, quite a few years later, about 10 years later, Doron Behar and his colleagues, 3 of whom are sitting in the audience, at least 3 that I know, Doron, our distinguished guest from Estonia, and Professor Karl Skoretsky, came up with a new study, much more refined, with many more chromosomes, many more analysis, and they came up with the conclusion that it does not represent the Kuzarim. And we'll hear a wonderful talk in a few minutes. So I think this is the classical way that science is going forward. We put up an hypothesis and someone rejects it. That's wonderful. Now there are new hypotheses to study and to reject maybe. And so I'm very happy about this study. And if I have a couple more minutes, I want to show you something else. By the way, this paper, this work that Doron will talk about, is an amazing example of excellent scientists from all over the world who collaborated in order to produce this beautiful story. And I wish some of our politicians would have learned to collaborate in such a way. Okay, this is a curiosity. About a few weeks ago, I think sometime in September, I received this strange, angry mail. Why do I say angry? Because it is bold letters, large letters. You don't see it in proportion to the other letters. And it was clear that the person whose name is Adil Attar was shouting at me. And he just encountered our paper from 2000, And he has mentioned that he has published the same, it says finding. Do you see the word finding? That doesn't matter. As a theory, in a very famous newspaper, which comes out in Los Angeles, and it's called Beirut Times. And why didn't we refer to his paper? It would be kind and appreciative to at least recognize the existence of such finding about 10 years before your research was conducted. Anyway, I apologized, and we had some correspondence, and it turns out that there are a group of Arabs, I don't know if only Arabs, but one of them wrote to me that there are a group of intellectuals who are meeting, who all work in the Los Angeles area, and they are engineers, construction, and all sorts of things. And they are meeting there and discussing all sorts of things. And they are open-minded. But I want to show you, he's also sent me the paper, so this is quite amazing. Palestinians, the lost Hebrew tribes. Never mind the confusion of history, but quite a strong argument. And his argument is based on himself. He was raised in an Arab country as a non-Arab, and he said that the Arabs, wherever they are, they Arabize the population. He came from Arabized Iraq. Maybe I'll go back and will be. And his conclusion was that 80% of the original Jews were converted. First, the being identified as Jews were converted to Christianity or later to Islam. Nearly 80% of the original Jews were converted, and today they are called Palestinians. And he concluded this article in a nice way, and he suggested that research should be conducted with mathematical models into what took place during Christianization and Islamization period of the area. You owe it to your heritage. So now we are going to hear about our heritage from Doron.
תודה רבה, אריאלה, for these nice and interesting remarks. And now I would like to ask Doron to deliver his talk.
The title of this presentation actually is labeled after the conclusion of our studies about the potential genetic link or affinity between Ashkenazi Jews and the previous kingdom called Khazaria. It is concluding that we cannot identify any genetic proof that such a link this existed. So I'm giving you the bottom line of this presentation. However, I would like to lead you to this through the coming presentation by first explaining to you about some review of genetics and principle of archaeogenetics, which is going to be something like half of the talk. And then the other half of the talk is going to look carefully into the Khazar hypothesis, and we will also be sharing with you some future aims that we are working on them currently. So we will have to first understand what is population genetics. So that is population genetics. There is some kind of a population here. There are different kinds of creatures, and all of them have white, long ears. And then one needs to understand if there is any connection to this population from one side and to this very naughty rabbit. So that is how we start to study about population genetics, and I'm emphasizing that this is a completely out of interest of simply seeing the variation inside our genome, which might mimic, might reassure, or might disprove what we think or thought before by our other disciplines that are studying populations. So a first review of genetics, and I do apologize if some of you already know it, I just want to bring everybody up to speed. So a review of genetics is that the entire genetic language is being written from 4 letters. only A, C, G, and T. And that is the genetic language. And as compared to Hebrew, which has much more letters, of course, genetics use only 4 letters to make all the huge variation that we see among humans today. These letters are being collapsed into books. And there are a total of 3 billion letters which are being arranged into books. So if each of these one is a letter, eventually they are each condensed into this book that geneticists are calling chromosomes, only to make the life of non-geneticists harder to understand it. But this is a book. Then, like any encyclopedia, the human genome is all of the chromosomes that are existing in each human genome, which is a total of 23 pairs of books. Why am I always saying pairs, pairs, pairs? Because 1 copy is coming from our father, and 1 copy is coming from our mother. So we have a total of 46 chromosomes, 23 times 2, plus another molecule that is called mitochondrial DNA, sometimes referred as the small genome. Many names were given to it, but this is not part of the chromosomes. Here is a picture of a chromosome that is of a human karyotype, showing all the chromosome in a given cell. And as you can see, there's chromosome 1 to 22. They are simply arranged by their size. And this is 1, so we have 1 from the father, 1 from the mother, and so on and so on. And the 23rd pair in male is XY, as you know, and in female is XX. Okay. Please make a distinction that the X chromosome is not the mitochondrial DNA. Okay, I'm preempting some misunderstanding that might occur because we are using the empty DNA to trace maternal ancestry. Now, so if we summarize, if there's 1 pair of sex chromosome, XX from the female, 1 X must come from the father and 1 from the mom, and XY, well, X comes from mom and Y comes from dad. We are going to see it very soon in graphic. And there are numerous copies of the mitochondrial DNA, which is this additional piece of genome that is circulating in our cells. Now, one of the major driving forces for the differences that we all see in each other is mutations. Mutation is not a bad word. Mutation doesn't mean that this is something which might be pathogenic or lethal. It's not like my first son is telling to my second son, you are a mutation. That's not what mutation is. Mutation is merely any change in the genetic material, not more than this. Now, it might indeed have lethal consequences. If it is causing disease, it might be completely silent. Here is an example of a mutation. You see that this is the one individual, this is the second individual. I told you that the entire language is A, C, T, and G. You can see here some order of these nucleotides in this particular region of the genome that we read. And you see that this individual has a C, but this individual has in the very same position a T. They are different between themselves. Now, this might be the entire difference between a healthy individual and an individual that is suffering from cystic fibrosis. But that can also be, for the ones who know how to look at it, the difference between an African and a Native American. Now, you are going to hear a lot of things, exactly the name that is describing this mutation, SNP, single nucleotide polymorphism. One nucleotide is changed. Only one letter is changed. I'm going to switch here for 1 minute to Hebrew so you can all understand it. If you have a long recipe to making bread, probably one of the lines will be 1 kilogram kemach. If somebody made an error and copied this line by an error and wrote 1 kilogram kerach, it will only be one mutation, but it will not be bread. Okay? So this is what we are looking for, and we can certainly also show that this difference may represent different populations. Okay. Now I want to try to take you into the ancestry, and how do we trace ancestry? How do we do phylogeography? How do we do archaeogenetics using the DNA which is in our body, which is the relic that we have inherited from our parents and great-great-great-parents before them. So this is a given individual, and we are looking to his line. In this particular line, we are looking to his male line. And the male line, of course, he has one father, which is here, and then this father had one father, and this father had one father, and this father had one father. And we have to ask ourselves if we can trace this paternal lineage somehow using genetic markers. So let's try to discuss what I've just showed you previously. If that is the mother, she has two X. We've already agreed about this. If this is the father, he has an X and a Y. That is, might be different X's, from different genetic pools. And when they breed, there might be a few options for the chromosomes to merge. This X can come with this X and have an XX. That will be a daughter. But there are more opportunities, of course. This X can come with this Y, this X can come with this X, or this X can come with this Y. And you can see beautifully why 50% of the born individuals, a child, are females, and why half are males. But let's do one small trick here, only change the colors, and this is what you see. So now it must be clear to you that the Y chromosome of any male in this world can come only from who? From his father. Now, in different Western societies, we tend to do one more thing that is passing from father to sons, which is the surnames. So, for example, we know that, for example, might be a situation where two individuals have the family name today of Shapira. They don't remember that they are related. But maybe, if both Shapiras are the sons of one ancestral Shapira 500 years ago, they will not remember. But who will remember it? Their Y chromosome. The same goes for Kohanim. The same goes for Leviim. And we are going to address these issues later in this presentation. But of course, you might immediately tell me, hey, this guy also has a mother. What about his mother? Can we trace the mother? Then, of course, we can trace the mother. That is, now we know that we can trace her. And how do we trace her? We trace the mother with a molecule called the mitochondrial DNA. The mitochondrial DNA is a very small piece of genome, which is external to the big genome. We said that the big genome is 6 billion nucleotides, 6 billion letters, 3 million from the father, 3 million from the mother. The mitochondrial DNA is a circular genome. It's not in chromosomes, and it is only 16,000 base pairs. It's very small compared to the genome. But it is being transmitted to us via uniparental mode of transmission, which means that only the mother will deliver it to her offspring, but only the female will keep on giving it to their next generation. Which means that if you are a male, you can trace the paternal lineage of your father's grandfather, great-grandfather, and so on using the Y chromosome, and you can trace your maternal ancestry via the mother, great-mother, etc., two lineages out of the many, many lineages making your ancestry. So this is only these two, and we didn't touch here all the ones that are not in direct ancestry of male and direct ancestry of female. Okay. We are going to do now a small experiment together, and I ask you to treat all the colors as one color. And we are checking now a given position in our genome, one of the 3 billion positions, and we simply check what occurs in different populations worldwide. So we see that in these populations there is a lot of T, just to say, and the color is C. We see here that there are many, many Cs. But we see here that there is no C, there is no color. So if I'm going to bring you now an individual, without you being population geneticist or any alike, and I will tell you, I have now a new individual, and this individual doesn't have a color. And I'm going to ask you, is he European? Most of you might say, no, he's not European because previously we have never seen in a European this color. And this is how population genetics start, very simply. Now, this is only one position out of 3 billion. Of course, we can start tracing these positions that are unique to different geographic locations, to West Eurasia, East Eurasia, to Sub-Sahara, etc., etc. And then we can try to identify regions in which there was admixture or source to each other because they have different positions that are showing different nucleotides. Now, that is very simple. We have done nothing complicated so far. It's simply that we have checked which colors are in which region in the world. And this is a bit more complex map. And you can see here something interesting. You can see here that different colors are in different places in the world. This is the paternal lineage. This is the maternal lineage. And you can see that, as always, and as is the news, the Middle East is full of mess, is full of colors. But you can also see something very interesting here. You can see that while in the Middle East there are many colors, the number of colors are being diminished as we go towards the America, until here in South America we see only one color for one Y chromosome. So you can actually see that populations in the world have different patterns of genetic Y chromosomes that are representing them. But then you might ask yourself again immediately, okay, I see this color, where else can I find it in the world? Maybe I can find it in Africa? No. Maybe I can find it in Europe? No. Maybe I can find it in Siberia and in East Eurasia? And you can actually see that the closer we get to the Americas, we can see more and more of this color. So that might trigger you to think, aha, maybe this color is some kind of movement of people coming into the America, not from Africa, but maybe through Beringia, which was the bridge between Asia and the Americas. So you can see that the fact that these colors are in different places in the world not only allows us to characterize different populations, but might also tell us something about potential source and migrations among humans. But then again, we need to speak about evolution a bit, because mutations don't just happen. And I want to introduce you to this concept by stealing this word from Terry Pratchett from Small Gods, who wrote that events just happen, one after another. They don't care who knows. But history, history is different. History has to be observed. Otherwise, it's not history. It's just, well, events happening one after another. So if we change it a little bit, and we change to mutational evolutions, we get the very same picture. Because mutations don't just pop up. They pop up on living individuals and all the genetic material that these individuals carried from their ancestors. We are going to see very soon how it is actually working. So, phylogenies. What are phylogenies? Phylogenies is our ability to draw a pedigree that coalesces different individuals living in our days. And let's see how it is working. Let's say that there is a book. There's a book. The book has 1,000 pages. It's full of letters, full of words, full of chapters. And that is the first edition. Now, the book was so good that it was sold out, and now they want to print a new version. So they go and they print a new version, but in page 352, in the third line, in the second word, in the first letter, they change from a C to G by arrow. Now we have two types of the book, correct? One book will be the first one, the ancestor, and the second book will be the descendant. Now the second version was also very good. It was sold out. They want to take the second book and copy it, and now they make a mistake in page 700. Ta-tam, ta-tam, ta-tam. It is obligatory that the mutation that was by error introduced into the second book is also in the third book, correct? This is how it goes on and on through the editions. Now again, geneticists have coined the word haplogroups to say different versions of books. Again, only to confuse the non-geneticists, but this is only versions of books. But now you should be asking me, okay, we understood that there is first book, second book, third book, but how do you know who was the ancestor and who was the descendant? How do you know which side it starts, right? And the only way to do it is to look to out species, to look to the closest species that are known to us today and see to which side they resemble more, this side or this side. And then we conclude that this is the beginning. Now, will you have an idea which is the closest Homo ancestor that we have DNA from and we can compare our genome to? Some of you might say chimpanzee or the pan, but no, we do have DNA from Homo neanderthalensis that indeed was distinct, is extinct now for 40,000 years, 30,000 years, but still some DNA we could have harvest and have some information from it. So this is how evolution goes ahead. And we can actually draw this tree. This is a tree of mitochondrial DNAs of the maternal lineage, and what we can say is that 200,000 years ago, all humans, all mitochondrial DNAs of all humans coalesce into one ancestral mother. In non-scientific literature, sometimes this ancestor is labeled as Eve, but that is, of course, not a name that we are coining. And then we can see that we have these two branches, and they are keep on splitting. It doesn't mean that here we didn't have more lineages. What does it say? That these lineages are extant, are still our days visible. Many others were simply extinct. Now, does any of you have an idea of what this color represents? So this color represents the total variation that is found outside of Africa. So that is clearly showing us that probably the evolution of Homo sapiens, of our species, started in Africa, and this is an exodus. Actually, I have to mark for you this line, which is roughly 70,000 years ago, which is the coalescence of the last successful exodus out of Africa. There might have been additional exodus previous to this, but this is the one that was successful. By saying successful, I mean that Homo sapiens spread throughout the world. So now we know that we can have different haplogroups, and we can suggest dating of different haplogroups. So we have the ability to show how potentially Homo sapiens migrated in the world. One of the roots, for example, is South Asia. It is called the fast train, or the fast boat, because the lineage here have very similar ages, very similar coalescence ages, which is giving us a hint that the movement was very fast along this coast. Now, please don't take these lines as physical lines that the people went through. It is also, of course, only schematic to explain how migration moved. Now you might ask me, okay, very nice. You can trace my paternal lineage, you can trace my maternal lineage, but you know something? There is a story in my family that my maternal great-grandfather, which you cannot trace, is of Native American ancestry. Can I trace it? It's not in the direct line of the mother, it's not on the direct line of the father. So I cannot trace it using the Y chromosome and mitochondrial DNA. But remember, I still have the entire genome. So let's look at the entire genome. The entire genome is made out of the 22 chromosomes. These chromosomes, not only that there are many of them, and that we can get by random a different copies of a different set of 22 from father and mother, they also have a very annoying characteristic, which is called recombination. What is recombination? During the meiotic event, they have the ability to switch part of them between sister chromosomes. So they are making things really hard. So we cannot trace on a chromosome, which is not Y chromosome, our ancestors, because they keep on switching regions between them, and also they can come in any combination during the meiotic event. However, not only that they are doing this, also each individual, by definition, must have many, many, many different ancestors in any given population. And here I should pose to you a very classic question, and I will simply ask you, most of the humans that ever lived in the world are lived when? 100,000 years ago, 10,000 years ago, 100 years ago, or today? Today. Today there are 7 billion people and growing. So that means that by obligation, all of us share hundreds or thousands of ancestors, that our genome is a fraction of all of them. Now, we want to try to trace it. So how do we trace it? This is a complicated figure, only to tell you that there is this method that was used very often using bead arrays, which are chips. This is actually a chip on which we can burn small molecular reactions, never mind the exact mechanism, that are looking to the SNPs, to the single nucleotide polymorphism that we have shown you. And from previous projects, the big ones of them are the HAPMAP project, we can see that we do find differences between different populations. And we can see that something like a 600,000 base pairs are found to differ between different populations, which means that in this population they have mainly C, in this population mainly T, and then in another one it might be G and A, and maybe there's a third one, which is this population is C, in another one is T, and then the third one is A. But we can burn these 600,000 base pairs. Today it's even more, it's almost 5 million of them you can do. We can burn them, and since they are coming from all the different places of the genome, they are going to give us some kind of a snapshot of the genome. Now, what do we know? If indeed populations evolved separately from each other, there were no airplanes, there were no boats 10,000 years ago, and the mountain was really a barrier, a river was a barrier. So if populations were really isolated geographically or culturally, then what is true about the ancestors? That they kept on marrying and shuffling the same genetic pool within them. So that means that we are expecting that such a large shot of the genome will also show some coalescence, some resemblance within the same population. Okay. I'm sorry if some of you have read the next 2 slides with me. We will summarize the part of understanding what is evolution and how it is progressing by reading it together. I'm going to waste 3 minutes of the precious time given to me in the presentation. Hopefully after this you will all understand how evolution is evolving and we can get to the data that we came for. So the European Commission has just announced an agreement whereby English will be the official language of the European Union rather than German, which was the other possibility. As part of the negotiations, the British government considered that English spelling had some room for improvement and has accepted a 5-year phase in plan that would become known as Euro-English. In the first year, S will replace the soft C. Certainly, this will make the civil servants jump with joy. The hard C will be dropped in favor of K. This should clear up confusion, and keyboards can have one less letter. There will be growing public enthusiasm in the second year when the troublesome PH will be replaced with F. This will make words like photograph 20% shorter. All true. In the third year, public acceptance of the new spelling can be expected to reach the stage where more complicated changes are possible. Governments will encourage the removal of double letters which have always been a deterrent to accurate spelling. Also, all will agree that the horrible mess of the silent E in the language is disgraceful and it should go away. By the fourth year, people will be respected to steps such as replacing TH with Z and W with V. During the fifth year, the unnecessary U can be dropped from words containing O, and after this fifth year, we will have a real sensible written style. There will be no more trouble or difficulties, and everyone will find it easy to understand each other. The dream of a united Europe will finally come true, and after the fifth year, we will all be speaking German like they wanted in the first place. Okay. So these small mutations, which seems to be very tiny, when they accumulate, they actually allow us to see different variation and different haplogroups and different additions within our ancestors. I'm going to have one slide on the Y chromosome, on the paternal ancestry of what that we think that is the best knowledge that we have on Y chromosome of Jewish populations. Nothing much was changed since this paper that was led by Professor Mike Hammer from Arizona and Batsheva Bonet Amir from Tel Aviv University. And what that you can see here, that triangles represent Jewish communities and the other ones are non-Jewish communities. And you can see that Ashkenazi Jews, Jews from Rome, Kurdish Jews, Yemenite Jews, are very close to Syrian non-Jews, Palestinians, Lebanese, and are relatively far from their host populations in the diaspora. Nothing much was changed since then of our big understanding of the situation. For example, I'm going to mention this one haplogroup. Again, I'm sorry for having to say J1EP58. It's just an addition in a book that is known to be the haplogroup to which the major Cohen model haplotype, Kohanim lineage, described by Karl Skoretsky, was actually found. And I just want to show you that this J1EP58 is actually found a lot in our region, and then again in Jews, and then in Kohanim, showing some link of Jewish populations to the Middle East. As you can see, in Europe it is very minimal, but in this region it's much larger. So what we do think, exactly as presented by Ariela a few minutes ago, that we do have a general understanding that at least a substantial amount of the paternal lineages of contemporary Jews are affiliated to the Near East. If we're going to move to the maternal side, again, only one slide and we move to the Khazar hypothesis. If we are going to look to Ashkenazi Jewry, then what that we see is that indeed something like 40% of contemporary Ashkenazi Jews can be traced back to 4 maternal lineages. And this is part of what was already suggested by Uri, that is the limited founders that have given, that are the founder effect, that is the reason maybe for the extensive number of genetic diseases in Ashkenazi Jews. So that is for the Ashkenazi Jews. For the non-Ashkenazi Jews, we see something very interesting, and we see that each population almost have a different founder maternal. So, for example, Georgian Jews will have a different maternal founder than Ashkenazi Jews, than Yemenite Jews, and so on. I don't have time to go into the details of this, but certainly how strong was the founder effect is different between the different populations of different Jewish communities. So, for example, you can be in a situation where in Georgian Jews, 56% of contemporary individuals are coalescing to one mother, and to the Moroccan Jews, in which you can't really find a very strong founding lineage. And here we step into the Khazar hypothesis, and we're going to start it by discussing what many papers were showing so far about a picture that is being shown about Ashkenazi Jews from looking with these chip arrays, with the 600,000 letters checked throughout the genome. What we can pretty clearly say is that the Ashkenazi Jews, in this particular analysis, which we'll elaborate on in the next slides, are taking an intermediate position between European populations and Middle Eastern populations, and that they completely overlap with Spanish Jews. Spanish Jews is meaning the Jews that directly descend from the Spanish exile, like Bulgarian Jews and Turkish Jews, and from North African Jews. So that is a pretty common figure that is being repeatedly shown by different authors, different groups in different years, and even recently using really whole genomes, not only the snapshots. And generally speaking, that is the picture. We can also see here that Iraqi Jews, Iranian Jews, Georgian Jews, and Azerbaijani Jews are making a second cluster, which is falling right on the Middle East. And here we see Yemeni Jews, which are creating their exact cluster, their specific cluster. So they are all very close to the Middle East. And Ashkenazi Jews are in this intermediary position. This is the vast majority of the Jewish communities. And we don't see here, for example, the Ethiopian Jews or the Indian communities, despite that for Indian Jews, there are some other links to the Middle East, to the Near East. They are far away on this graph, so we can't see them here. Now, some individuals have, throughout the years, some scientists throughout the years, including us, have also raised other possibilities for explaining where Ashkenazi Jews are coming. Of course, this is more suitable, more compatible with a particular historical theory claiming that the Jewish communities of East Europe, the Ashkenazi community, was created by a series of bottlenecks starting from the Levant, going to Rome, North Rome, then to the Rhine Valley, and then to East Europe. So that is, of course, more suitable, more compatible with this theory. But there is another theory that is the Khazar hypothesis. You all know what is the Khazar hypothesis because you are sitting here because of the title of the lecture. But what the Khazar hypothesis is claiming is that the previous historical route which I described is actually wrong or is minor, and most Jews are coming from a conversion of individuals, of a population called Khazari that combined. It was a conglomerate of Nordic tribes, of nomadic tribes that were Turkic speaking, that were living in this region somewhere around 700 to 900 to the Christian count. And they have converted to Judaism, and they have moved to Europe, and this is why Jews are of Khazarian, Ashkenazi Jews in particular, in specific, are of Khazarian ancestry. Now, this theory, of course, captured also the eyes of geneticists, and maybe one of the first examples is indeed the story of the Ashkenazi Levites, which Ariela already detailed a bit about her paper, and also our paper from similar time frame was discussing the very same one. And this is actually a story of a haplogroup called R1A, yet another edition of the book, that we simply checked it in Ashkenazi Cohen, Sfaradi Cohen, Ashkenazi Levites, Sfaradi Levites, Ashkenazi Israel, Sephardic Israel, I mean Israel meaning not Cohen and not Levi, and then in non-Jews, Germans, Norwegians, Sorbs, and Belarusian. And what you can see is that astonishingly, for Ashkenazi Levites, 51% belong to this R1A haplogroup. Very interesting. While in other Jewish communities, in Ashkenazi Cohen, it's in 1%, in Sephardi Cohen, 5%, in Sephardi, 3%, but in non-Jews, it is 12%, 21%, 63%, 50%. So of course, that have led us to try to set the origin of these Ashkenazi Levites, and we actually wrote that one attractive source would be the Khazarian kingdom, whose ruling class is thought to have converted to Judaism in the 8th or 9th century. So I think that was my first paper in population genetics. Naturally, I was very excited to maybe suggest something like this. And of course, as Ariela told you, later study, which was published last year, showed different results, and we are going to get to it at the end of the presentation. Other studies also tested it. And they detected a small but measurable signal of similarity between Ashkenazi Jews and the sample of the Adigei population from the North Caucasus region. So these are different papers, all showing this, and all, of course, thinking about the potential Khazar ancestry for Ashkenazi Jews. But there was really a paucity of data. So then came a paper which was entitled The Missing Link of Jewish European Ancestry, contrasting the Rhineland and Khazarian hypotheses, in which the author actually suggested very strongly that there is a clear-cut proof that the majority of contemporary Ashkenazi Jews rely to Khazarian ancestry. And to summarize the study, Palestinians were considered proto-Judeans, which means that Palestinians were considered the ancestral population to Jews. That is, of course, not a good terminology to say proto, because Palestinians are not proto-contemporary people, but nevertheless, it's a good, maybe a good proxy for the Levant. By the way, samples were from our study, so this is why we are very familiar with the data. But then it was also mentioned that Caucasus Georgians and Armenians were considered Proto-Khazars. That is a bit more provocative already, because we have to actually understand if that has real basics to claim that Georgian and Armenians are Proto-Khazarians. And then it was calculated that the genetic distance between Armenian and Georgian population and Ashkenazi Jews is small. So the conclusion was that Ashkenazi Jews are of Khazarian origin. Now, of course, we have to treat it in a very scientific way and check our data again. And the first thing that we have to do is, of course, take more data from the, I will show from the Caucasus. But even before coming to the scientific data, I must go through some very important principles of population genetics. The first of which will be self-identification. Population geneticists start their study by going to the individual and asking him, how do you self-identify? How do you self-affiliate? And we are not changing what this individual is saying. So this individual is saying in Newsweek that he thinks that he's affiliated with Professor Karl Skoretsky sitting in the audience now. Now, they actually both self-identify as Cohen priests. Now, Professor Skoretsky says, really? And goes to check it. And he says, okay, if he says he's a Cohen, and that's a surname passing through generation, and I'm saying I'm a Cohen, and this is passing through generation, maybe our Y chromosome remembers it. So there's come studies of the Jewish priests of 97 and 98, and actually it is becoming apparent that these two gentlemen are indeed belonging to a very specific haplogroup. That was the entire knowledge we had in these times about phylogeny, and they indeed fall, both of them, here. Still 10 years afterwards, the study is repeated with a much larger phylogeny, and the conclusions remain the same. Now, that means that these two individuals, which phenotypically seems to be very different, in one aspect of the genome and one aspect of self-identity, they overlap. Now, this is very important to explain here something. All the geneticists are doing, they are taking a group of 100 individuals that all say, we are Kohanim. We are Jew. This is the way they define themselves. Then they take a group of 100 people who are saying that they are not Kohanim. And then they check the Y chromosomes. And they see that for the 100 people that are not Kohanim, that's saying that we are not Kohanim, they have 100 different editions of the Y chromosome. But when you look at the Kohanim, you see that 50 of them have the same edition. And then 25 more have a second edition, and so on. So what that we have said now, that 50% of contemporary Kohanim share the same paternal father. Now, can I, as a population geneticist, go back to the other 50 and say, you are not really Kohanim? No. Okay? All that we are doing is learning genetically if there is a connection between the oral tradition, or oral story of these individuals and the genetic findings. We cannot take it back to the population and say to somebody, you know something, you are not really Native American. You are not really, that's what we can do. Okay. Then, of course, comes the question of who is a Jew. That is a very important study, a demographic study, which was published by the Pew Institute that they studied in the US how Jews are self-affiliating, and they do recognize that who is a Jew is an ancient question with no single timeless answer. And they actually show what was the way they were thinking. So they were first asking people if they are Jewish by religion, because Jewish is a strange word, because Jewish is an ethno-religious group, like Druze people. While, for example, Arabs can be Christians or Muslims. So they first ask if they are Jews or not. So some people say yes, and that they are Jews by religion. Some people say no, we are not religious, but we are Jews via ancestry. So indeed the question of Jews, of who is a Jew, is something we are not going to resolve among us, but it has many different answers. Some people say that way, and some people say that way. We are not entering into this, but the only thing that we know, that when we give our questionnaire to people, they are writing down that their ethnicity or religion is Jewish. This is how they self-identify. It doesn't mean that we have a proven fact that they are indeed the descendants of the Hebrews, but this is what they say today that they are. But who is Khazar? There is no contemporary population, and we have in the database of the Estonian Biocenter all of these populations coming from the Caucasus. None of these populations self-identify as Khazars. None of these populations are saying, yes, we know that they are descendants, and there is no historians who can point up to one particular one which is descendant of Khazars. And you can try to look for it, for example, by which language they speak. So for example, since the Khazarian people are known to speak Turkic languages, then maybe Kumiks might be some representative. Maybe Nogais, despite the fact that Nogais came to the Caucasus 200 years after the Khazarian kingdom was eliminated, disappeared. But so there is really no one that can say who are the descendants of the Khazars. So the only thing that is important to say here with respect to the comparison between the Jewish and the Khazar is that the situation of identify a Jewish descendants or people who self-identify as Jews is certainly not worse than to try to identify who are Khazars, because at least the Jews are saying we are self-affiliating as Jews. Maybe it's not true that they keep this, that they maintain the lineage for 2,000 years, but there is no one saying we are Khazar descendants. So without knowing who is really Khazar descendants, we had to actually sample from all of these populations throughout the Caucasus region to try and see if there is any connection between Ashkenazi Jews and Khazarians. So this particular study took 1,077 forced samples, which is a very large number. Ashkenazi Jews were 99 out of 202 Jews. And then we took all of these populations from the Caucasus, and we also took two populations, the Chuvash and the Tatar, because Chuvash are sometimes noticed as potential descendants of the same Turkic language speakers. And we took them, so they are known to the Caucasus, but we included them as well. And of course, we could have put in much more Ashkenazi Jews, but you are not allowed to have too much difference in the numbers of samples belonging to one population, because that might create some bias in the data. So this is why the numbers are kept relatively even. And actually, it is covering what we think was previously the Khazarian kingdom. So we have Georgian and Armenians here. We have from the real major portion of the Khazarian kingdom, and close to Atil, which was the capital. And then we have even Chuvash and Tatars. These are more European samples, so they are not mentioned here. The rest of the samples were, of course, from Europe, from Africa, from Sub-Sahara, from North Africa, from Asia, from the Middle East, a lot of them, and so on and so on. And we started to do some analysis. This is principal component analysis. I'm sorry it's a bit blurry, so I will try to clarify to you this. So what we keep on saying, the principal component analysis, is that always the population of the world are being defined in the first axis. Principal component analysis is saying, show me the two axes. If we are comparing PC1 and PC2, there are endless PCs. There are endless vectors that we can try to look for connection between two phenomena. In this case, it's human populations. African and not African populations are being split, and then east to west. You can see it better here. So Africa and non-Africa is the best, is first, and then west to east in the world. And I've marked here for you the Jewish populations in red. So here you can probably understand that you can find Ethiopians, here you can find the Indian Jews, and here the overwhelming bulk of Jewish communities are falling here in a region which is populated with Middle Eastern and European and Caucasus population. Now, this is an enlargement of this particular piece, and each of these circles represent the median vector of all individuals in a very population. This is Ashkenazi Jews. As you can see, this is Armenian Jews. This is Armenian non-Jews. This is Georgian non-Jews. And this is North Caucasus. You can clearly see that Ashkenazi Jews are closer to other Jewish communities, which are in red. And you can also see that Armenian are very close to different Jewish communities, also to South Italians, Sicilians specifically. And you can see that actually North Caucasus are very far away. So if one wants to say that Armenian and Georgians are ancestral to Ashkenazi Jews because they are close to each other, then by definition he must also say that they are ancestral to Georgian Jews, to Iraqi Jews, and to Italians and to Sicilians. If we take another analysis, this time is localization analysis. I see that some of the slide is cut, I'm sorry. What is localization analysis? This time you take half of your samples of populations you know where they are geographically. You know that Palestinians are in this region. You know that Druze are in these regions. And you tell the algorithm, this is where these populations are. And then you take the other half of the population, and populations you don't know where they are coming from, like Jews, and you try to put them on a map. And again, what we see is Ashkenazi Jews here. And the Ashkenazi Jews are closer to Sicilian Jews. This is Cypriots, non-Jews. These are all Jewish populations. I have marked again Armenian and Georgians, and I have marked North Caucasus. Again, what we see is that Armenians are actually very close to different Jewish and Levantine populations which are here. And actually, Armenians are closer to Cypriots than to Ashkenazi Jews. So what we start seeing is that maybe Armenian and Georgians are simply close to Near Eastern populations because they are geographically very close to them. And it doesn't say anything in addition. So we go on to this analysis called admixture. Admixture says the following. There are, dear computer, there's an algorithm inside that people were working on. Please divide the world for us into 10 different ancestral groups. And we don't tell the computer which individual comes from which population. And what you can see is that Africans are being clustered here in two different colors. Then we have the Middle East, then we have Europe, then we have Caucasus, and then we have the East Asian population. And you see that there are colors that are specific to different places in the world, but you see that in many populations, it represents the complex human demography, evolutionary demography. And you see that there are some colors that are representing a region and also shared between regions. If we are going to look to the Jews, here, the Jewish individuals, we can see that they fall quite close to Druze, to Samaritans, to Cypriots, to Lebanese, to Syrians, and so on. And if we look to Caucasus, we look that they have similar colors, but there is something very important. This particular color, which appears primarily in the Middle East, appears only in Armenian and Azeri populations. Again saying that maybe the population that was chosen to be as a Proto-Khazar population is not really the right one, because it is simply one that is geographically in proxy to Middle Eastern population. And we can also see that there is no real difference between Samaritan Druze and Sephardi Jews. So if we again think that these populations are ancestral to Ashkenazi Jews, then of course one must say that they are also ancestral to Druze and Lebanese and so on. Here is a more accurate, numerically accurate analysis. And what we see here is that each of these graphs represent a different population which is listed here. And we are asking Ashkenazi Jews, Cypriots, Druze, and Palestinians to which population they are closest with an allele sharing distance. And this green line, this green line, this green line, and this green line are South Caucasus population. Again, we see that there is no uniqueness to the proximity of Ashkenazi Jews to Caucasus population, and actually Cypriots and Druze might actually be even closer. Okay, one of the last slides that are going to be complicated. Identity by descent is a method which is checking only proximity in the last 10 generations, let's say. Maybe a bit more, maybe 15, maybe 20, but very close. And what we see here that Ashkenazi Jews are closest to Sephardic Jews. I think that, oh, here it is. And then to North African Jews, and then to Eastern European and Western European, and then to all the rest. Again, not a particular recent connection with Caucasus, but of course this is not expected because the time of the Khazarian kingdom predates the ability of this methodology to predict recent ancestry. Here is another paper from a Lebanese group actually showing again that Armenians are actually closer to Cypriots in a population tree, and Sephardic Jews and Ashkenazi Jews very close to each other, and again the same conclusion in another paper. Another paper published recently by Lazardiris et al. Was trying to estimate by a model, a very basic model which must be improved, what is the proportion of early European farmers, which are Near Eastern agriculturists, what is the proportion of ancient North Eurasians and of West European hunter-gatherer to the contemporary gene pool of European by comparing contemporary people to information from ancient DNA, from skeletons 7,000 to 8,000 years ago. And you can see that Ashkenazi Jews have 93% early European farmers as compared to much lower numbers for other populations. We go back to the Levite story, and that is the information that was not all published. For years, people were trying to find branches of R1A1, which is the edition of the book that Levites fall inside. And this paper was checking over 2,000 samples and didn't find any good internal structure. The resolution came only when we were able to actually start doing whole Y chromosome genotyping. Take the entire Y chromosome, 60 million bases, and run them. It's, of course, less in actual calculation. It's like 10 but this is what is called whole. And this marker, M5A2, was found to be in all Ashkenazi Levites. And I'm going to guide you through this. Ashkenazi Levites, 600 were checked. Of them, 13.3% were haplogroup R1A, the founder effect that Ariella was telling you about a few minutes ago. In non-Ashkenazi Jews, much less. It's 1.2%. However, in Eastern Europe, which we checked over 2,000 samples, 0 had this haplogroup. We thought initially, of course, there is 60% R1A in East Europe. There is 60% Ashkenazi Jews. It means that this is from the same ancestral pool. No, 0. In Central and Southern Europe, 2. In Caucasus, 1 out of over 2,000 samples. But in the Middle East, suddenly some proportion, 18 samples, and we can see that they are falling specifically in the region of Iran and the Kurds. And now I can tell you that we are also familiar with additional lineages that are not yet published from our region. So this is a global whole Y ancestry. It is 500 individuals taken from all of these individuals in all of these locations, submitted by a group led by Carmen et al. From Tartu, Estonia. And it's, of course, very hard for you to understand, but what I want to show you is that roughly 50,000 years ago starts some kind of a population of Y chromosomes. Y lineages burst. There are becoming to be much more Y chromosomes variants in the world. Now we have to allocate the Jewish samples inside them. It's not easy. As you can see, the red ones are the Jewish samples. So I'm going to help you a bit here. This is M5A2. This is R1A haplogroup. And you can see here that there's a root, and it splits into 2. And you see that here you have Ukrainians, Hungarian, you have Swede, Sami, etc., Russians, and there is indeed one Ashkenazi Jew among them, maybe suggesting a European introgression into the Jewish community. But you see that the founder effect of the Ashkenazi Jews falls into a different branch, which is close to Assyrian, Iraqi, Sugnam, Iranian Jews, Azerbaijani, Mumbai, Kirkit, etc., which seems to be Iraqi, India, which seems to be a more West Eurasian branch. We also can see here that the calculation here is that the age is roughly 1,600 to 800 years. So this level of resolution also allows us to give much better understanding of the origin of lineages. And here is another one that I have to show you, the J1, because the J1 is the one that is including inside it the Cohen lineage, so to say. And you can see here one very important thing. All of these samples here, sorry, this is not, the red is, can you see it here? All of these ones are sharing ancestry like 20,000 years ago, very close to this one, and J2 even before. And these dates are far before somebody knew what is Jews, what is Arabs. Of course, it's really almost almost global populations. And you can see that Ashkenazi Jews fall in very, very close proximity to Armenian, to Muslims Arab, to Saudi Arabian, etc., etc. Again, reconfirming the same notion that at least a substantial proportion of Ashkenazi Jews are of Levantine origin or Near Eastern origin. The conclusion does that Ashkenazi Jews share the greatest genetic ancestry with other Jewish populations and among non-Jewish populations with groups from Europe and the Middle East. No particular similarity of Ashkenazi Jews with population from the Caucasus is evident, particularly with the population that most closely represent the Khazar region. Ashkenazi Jews derive their ancestry primarily from populations of the Middle East and Europe, that they possess considerable shared ancestry with other Jewish populations, and that there is no indication of a significant genetic contribution either from within or from north to the Caucasus region. I want to, of course, mention again and clarify to you. Currently, we don't see any substantial or any significant clear link between Ashkenazi Jews and Caucasus population, which is different from the link of the same population to other population of the Near East or the Levant. Maybe in 5 or 10 years, the methodology will become even more robust, and maybe we will be able to find 1 or 2% or 3% that we currently cannot see. So you always have to be cautious in science. But today, to our days, we have no significant, we cannot detect any visible, well-documented connection between Caucasus population, presumably representing Khazar origin, and Jewish and contemporary Ashkenazi Jews. And with this, I want to thank all of our collaborators, really from many different locations. Of course, the first from Tartu, Estonia, then many others from Stanford, Tucson, from Armenia, from Russia, Italy, Spain, Sarajevo, Thessaloniki, and of course from Israel, Tel Aviv, and the Rambam Medical Center in Haifa. And with this, I want to thank you. And if there are any questions, please, if we are allowing, then we can of course take.
Dורון, for a fascinating lecture. And I'll take a few questions if there are. Yadin?
There is some historical evidence, I'm not sure about the fossils, that the work of
Shlomo.
Thanks.
As far as I know, there is some historical evidence for migration of a small population from the Khazar region to the Kiev region. Okay. Now, if this is the case, and this is based only on history and not on genetics, would you expect to find some characteristics of Khazar origin in that will give you some indication of what SPS you should try to find?
I should first excuse myself because I'm not an historian.
Neither am I.
So that will be not, I mean, it will be dangerous for me to answer it. Naturally, there is a great interest about this region of Kiev and maybe even the Krim region and some Jewish ancestry, Karaites ancestry, which is well discussed. However, this specific work didn't check it. I feel not very comfortable to discuss about history and about genetic data that we have not possessed and that we have not studied yet. And I might suggest that maybe here, maybe now or later, you might approach Richard Williams, who is, I think, a bit more familiar with this subject, and he can give you some more answers about it. So I'm sorry for...
טוב, אני יודע, אני גם מתחיל מהיסטוריה ואני אגיע לגנטיקה, כן. אני יודע שיש ויכוח בין היסטוריונים לגבי מה היה גודל ההתגיירות בממלכת קזריה. אז יש את הספר של פולק מתחילת שנות ה-40, שהוא הניח שאחרי המלך והשכבה השלטת, כל העממים השונים שהיו בקזריה התגיירו, וכך נוצרה אוכלוסייה מאוד גדולה שאחר כך, הרי שהם הפסידו שם לנסיך מקייב הם התפזרו, וכך הוא הגיע למסקנה באמת שרוב אוכלוסיית מזרח אירופה מוצאה משם. אז אני מבין שזה לא כל כך מתאים לגנטיקה. מצד שני יש גישה אחרת שאומרת, וקשה לדעת היום באמת מי צודק, שהמלך התגייר והשכבה השלטת התגיירה, ופחות או יותר זה נגמר בזה. והם אחר כך באמת נשארו נאמנים. והתמזגו בעם היהודי, כפי שגם מוזכר באיזה רומן על התקופה, גם כן, שבעמוד האחרון כתוב, הם לא עבדו לנו, הם התמזגו בינינו, ובכנס קודם הזכירו שלטורקיה, לקושטא הגיעו כוזרים שהתמזגו שם יפה. אז מה שאני מנסה בעצם לשאול, האם יכול להיות שבאמת הקבוצה הקטנה הזאת, שאולי לא השאירה סימנים בכל האוכלוסיות האחרות שציינת, פשוט התמזגה, וגם פרופסור אופנהיים הזכירה את זה בכנס הקודם וגם היום, פשוט התמזגה, ופשוט אתם לא מוצאים בעמים של היום דמיון לזה, שכך אין לכם פשוט קבוצת התייחסות.
בהחלט, אז זה בדיוק ככה, אני יכול לענות באנגלית?
כן.
So this is certainly, so the question only for Professor Williams was that there are the two theories that are saying that maybe a massive amount of Khazarians were converted into Judaism versus another historian theory saying that only the upper layer of the kings and their immediate proximities, proximal family members or clerks and so on, converted to Judaism. And maybe these small groups certainly did it, but we can't identify it in our genomic methodology. And certainly, that is exactly how I finished my presentation, saying that what we can say is that we cannot detect a significant Khazarian signal in contemporary Ashkenazi Jews. It is certainly possible that this event, this demographic event has happened and has never left a genetic signature in Ashkenazi Jews or have left something that we cannot detect today. But it is what I can say as a molecular geneticist clearly is that it is not a big portion of the Ashkenazi Jews. If we are going to find something like this, it's going to be something which today is at the level of noise. Okay, that's what we are saying.
Okay, Carl, you want that? שאלה אחרונה. Okay, עוד שאלה אחת ודי.
Okay. Don't even though you attributed the Hebrew proficiency to me, I'll permit myself to speak in English, if you don't mind. Will you comment on the four, because it does relate to the origins of Ashkenazi Jews. You mentioned in the middle of your talk the four founding maternal mtDNA lineages, but you didn't speak about their provenance. You didn't speak about their geographic origin, which could relate to this overall question and certainly is a point of discussion. Are you willing to speak about that now? And very much related or somewhat related is the use of Levant and Mediterranean, which one is tighter than the other, and are you willing to be more specific?
There is a very, so let's start with the second question, because that's an endless confusion that is in the literature. There is the Far East. Everybody knows where is the Far East. Then there is Middle East, and there is Near East. And there is Levant, and there is Mediterranean. So these are not separate geographic regions. Many times they are simply overlapping or one completely within the other. So that is actually one of the first questions I was asking. And again, Professor Wilms is not being a population geneticist, he is a population expert. We discussed it many times. And Levant is considered to be Israel, Lebanon, part of Syria, and then Cyprus is included within the classical Levant. Near East is a bit wider. The Middle East is anything from Turkey to Iran and Afghanistan to North Africa. Mediterranean is, of course, the Mediterranean basin, which is the northern and southern coast, and, of course, the western coast, which is the eastern coast, which is the part of the Levant. And something very important to say here is that today we say that Italy is in Europe, and in our mind, Italy affiliates with Sweden more than it affiliates with Syria or Lebanon. But maybe 5,000 years ago, it was very easy to go to Lebanon and to the land of Israel rather than crossing the Alps and going to Sweden. So this is a bit vague. What we are trying to narrow our discussions is to the Levant and to the Near East. We try to use less Middle East, and when we say Mediterranean, we clearly say Southern European Mediterranean, Northern African Mediterranean, or the land of Israel. So there is some confusion here, and it's certainly something that has to be corrected and be more, more robustly used in the literature by correct naming. As far as the maternal lineages of the Ashkenazi Jews, so there are 4 maternal lineages. There is no discussion about this anymore in the literature. Initially, there was discussion about this a bit, but now it is clear that roughly 40% of contemporary Ashkenazi Jews derive their maternal ancestry from 4 mothers, real mothers, not statistical ones, that lived something like, I'm just throwing numbers, 2,000 years ago, but some of them are younger, some of them might be a bit older. And there is now a debate in the literature, which was raised by a group in the UK claiming that they can prove that all of the maternal lineages are coming specifically from the Italian peninsula, as opposed to what we have suggested a few years ago, that the circumstantial evidence clearly suggests that one of them is from the Near East. It's called N1b. And the 3 others, the proof is not that so strong, but the circumstances are such that we think that they are from the greater Mediterranean. This is why we preferred a Levantine origin. There was yet a 3rd paper that refuted what the new paper said about Italy and said that the conclusions are actually opposite and they are coming from the Levant. We as a group, we didn't publish it yet. It takes time to write and to publish and to convince the reviewers. We still hold strongly to our original opinion, and we clearly see some critical phylogenetic errors in the paper suggesting that the maternal mothers are from Italy, and hopefully we are going to make it published and available in the coming months, which in science always become coming 1 or 2 years, but we are working on it. So that's what I... Yeah.
One last question.
המאמץ שעשית בעצם מראה שעשרות רבות של שנים ששני פרופסורים מכובדים, סתם דוגמה, אבל אני יכול לקחת כל אוניברסיטה בעולם כדוגמה, ובמקרה הזה פרופסור סטון ופרופסור לרנר, קיבלו משכורת מהאוניברסיטה העברית מיותר לחלוטין. במילים אחרות, כל אדם שעוסק בקווקז בצורה רצינית, יגיד מלכתחילה שההשערה שגיאורגים וארמנים הם אוכלוסייה פרוטו-כוזרית, זה... אני לא רוצה להתבטא, כן? זה אבסורד. וכבר נכתב על זה, אני לא מחדש כאן. העובדה שדבר כזה יתקבל לכתב עת מוביל, מעלה שאלות גם על הסוגה כולה. והשאלות האלה, במיוחד באקדמיה למדעים, דורשות קצת יותר שיח בין אנשי הגנטיקה של אוכלוסיות, שהם אנשים עם המון כוח. סליחה שאני אומר את זה, אני אומר את זה מתוך הערכה. ועושים דברים מדהימים, לבין אנשים ממקומות אחרים באקדמיה למדעים. עכשיו אני אתן עוד כמה דוגמאות מתוך המחקר שלך שהן שוליות, אבל מעלות שאלות. כשאתה מראה לנו רשימה שלמה של עמים קווקזי, ובעיקר צפון קווקזי, ומראה לכל אחד מה מצאתם, אתה לא מציין שמדובר בקטגוריות סובייטיות, שחלק מהעמים האלה בעצם שייכים לעם אחד. אם תראה את זה, אני יכול לתת כאן הרצאה על האתנוגרפיה והפוליטיקה של האתנוגרפיה של צפון קווקז, אבל זה לא חשוב. כשאתה אומר לנו שערבים נוצרים וערבים מוסלמים זה דבר אחד, אבל מצד שני דרוזים ויהודים זה קבוצות אתנו-דתיות, זה היה משהו כזה שאמרת, זה מעלה שאלה, כיוון שההגדרה מהי ערבי בעיני אנשים ממדעי החברה ומדעי הרוח היא הרבה יותר אלסטית, ומי שמכיר את ההיסטוריה של לבנון גם יודע שמשפחות מאוד דומיננטיות זזו בין הזהות הדרוזית לבין הזהות המרונית, למשל, בהיסטוריה הספציפית של לבנון. כשאתה עושה הפרדה בין אשכנזים לבין ספרדים, אבל לעומת זאת הקטגוריה ארמנים מופיעה כקטגוריה אחת, למרות שישנם הפרדות דומות בקרב הארמנים, כל השאלות האלה מעלות שאלות לא פשוטות על הסוגה. עכשיו, כידוע לך, ישנם אנשים במדעי החברה שכתבו כל מיני דברים מאוד ביקורתיים. בניגוד לזה, אני חושב שהגיע הזמן ליצור דיאלוג יותר משמעותי על הדבר הזה, וההערה שלי היא בעיקר לאקדמיה למדעים. חסר כאן גם היסטוריון שיתעסק עם זה, וגם אולי איש שמתעסק עם קטגוריות.
אני רק רוצה להעיר שלפני כ3 שנים היה לנו פה כנס שבו גם דורון הרצה, אבל היו אז גם היסטוריונים, זאת אומרת היה ממש דיון בין 3 אנשים שכל אחד הציג פן אחר פן אחר ושאלות אחרות ותשובות אחרות לשאלה שאתה מעלה עכשיו, אבל הפעם זה לא היה פאנל של דיון, אלא הרצאה של דורון שבאנו לשמוע.
כמובן הרצאה מרתקת ותודה, סליחה אם נשמעתי ביקורתי.
לא נשמעת ביקורתי בכלל, הפוך. קודם כל, אני חושב שההערה שלך היא חשובה ביותר. אני רק אתקן בהתחלה, כמובן שאנחנו מודעים היטב לעובדה שיש תתי קטגוריות בתוך, sorry, I'll go back to English, sorry. Of course, we are well aware of the fact that there are, to each region, there are sub-categories. Naturally, we don't have currently the time to explain exactly about each population, which is clearly something that if you're an expert in this field, you really feel that it is very important. We completely agree with you that speaking with historians, and there were 4 historians that were here, not 3 in 3 years ago, that all agreed to what you're saying, and we spoke with many more historians, and as you see, part of the authors of our paper are from Armenia. And so with respect to your first comment, we completely agree with it. There is one more thing that I must say here, and maybe I'm speaking in the name of Professor Williams, who taught me to say this sentence. He taught me to say that we as molecular geneticists, we are not doing our studies to please or not to please historians. We are out of agenda. We just try to say what we see in the data. And it is very interesting for us to speak with linguistics, to speak with historians, because genetics is not the absolute truth. Genetics is one of the disciplines used to study a population history. So clearly that is the situation. And certainly, with respect to your comment about how do we define Arab Christian or Arab Muslims, then naturally, I have personally many hours of speaking with people in different villages, in Israel and learning the ancestry of how these people arrived to the region. I'm not an historian, so I will avoid from speaking about these issues so that I will not, make terrible mistakes that even when I'm being filmed. So I will not speak about history, but I'm 100% sure that inclusion of more opinions from experts in the regions like you will only make our study even better. And if you would have told me the opposite from what you said, if you would have told me now, listen, it's very nice all the population that you have checked, but you didn't check this population, then my immediate reaction will be, if we can sample them, let's include them and check them as well. So this is where we stand.
תודה רבה. I would like to conclude this evening by thanking you, Doron, again for a fascinating lecture. And I would also like to thank the other speaker, who is Elickson and Ariel Oppenheim, and of course, Professor Richard Williams for the kind words and interesting words. And I hope that we have all enjoyed this evening. So thank you, everybody, and good night.
Thank you.
Goodbye.